Gene mutation classification, an essential understanding. Today we'll explore gene mutation classification. Why are they essential? And their real-world examples. Understanding these classifications help us grasp how mutations affect genes, proteins, and biological functions. These insights are crucial for the fields like medicine, genetics, and biotechnologies. What are gene mutations? They're permanent changes in DNA sequences in genes. It's important to note that mutations can affect gene expression, protein function, and biological process. What does this look like? Changes in DNA don't only happen in one way. It happens in multiple ways. And I will be talking about 3 examples of gene mutations that give an understanding of how these sequences are affected. The first one is substitution point mutation. One nucleotide is replaced by another. We can see this in the example where the ATGCC sequence is affected and one of the nucleotides is replaced by another, which is the G and A, which is a 1-point substitution in the nucleotide sequence. An example of this is sickle cell anemia. The HBB gene changes all the sequences with A to a T substitution. The second one is insertion, where adding one or more nucleotides to the sequence. We can see this in the example, the second row, where ATGACC is the new mutation of insertion because it's been added from the original one. Next to the G, it's adding an extra A nucleotide, causing the mutation. An example of this is Huntington's disease, where the CAG repeats in its HTT gene. And the last one I'll be talking about is deletion, where it's the removal of one or more nucleotides. In this example, we can see it as the first one, where the ATGCC before the mutation gets deleted and we are missing the G nucleotide in this example, causing the sequence to have a point where there isn't a nucleotide. And let's see what happens. What are the effects of mutations on proteins? Missense mutations change a single amino acid. An example of this is achondroplasia, where the GFR3 gene is changed in one amino acid sequence. A nonsense mutation creates a stop codon, which basically tells the sequence to stop and basically cuts it short. We can see this in an example of Duchenne muscular dystrophy, where the MD gene is cut short. A silent mutation has no change in protein sequence. An example of this, it may change or affect the mRNA stability, but there shouldn't be a change in protein sequence. And a frame shift mutation alters the reading frame of a codon. An example of this is the Tay-Sachs disease, where the hexa gene is altered when reading. Why are classifications important? Let's go back and think about mutations. Mutations refer to any change in the nucleotide sequence as a result of failure of the system to revert a change. The main categories are the type of DNA change, effects on protein, mechanism of origin, and effects on function. One of the classifications of gene mutations is spontaneous mutation, where an error during DNA replication occurs. In this example, I'm using the TP53 mutation in cancer. If you look at the diagram, you see that the TP protein is highlighted in green, and we can see like two different changes in the protein, what is attaching to, and we'll get into that in a second. But I want to explain what the TP53 gene does first, so we understand why these mutations are crucial to our understanding in the real world. It is crucial for tumor suppressors. It suppresses tumor genes that play a key role in regulatory cell division and preventing cancer. When mutated, it can lead to uncontrolled cell growth and cancer development. In a normal TP53 gene function, the TP53 encodes the P53 protein, often called the guardian of the genome. The P53 protein detects DNA damage or cellular stress. And with this, it halts the cell cycle to allow for DNA repair, triggers apoptosis, which is programmed self-death, telling the cell to kill itself, basically, to preserve other cells around it if the damage is irreparable. And it activates a transcription of genes involved in the cell cycle, regulation of DNA repair. The TP53 mutation can occur due to factors like UV radiation, carcinogens, or inherited genetic changes. These mutations often affect the DNA binding domain, preventing the P53 from activating in its target gene. When it loses its functions, in this mutation renders the P53 ineffective. As a result, the cells are damaged, DNA continues to divide, apoptosis is not triggered, allowing damaged cells to survive. DNA repairs mechanisms are not initiated effectively when these functions are lost. In some cases, certain TP53 mutations can result in gain of function. These mutant proteins might acquire new functions that promote cancer progression, such as increasing invasion, metastasizing, and resisting therapy. Without the function of the P53 protein, the cell cycle checklists, the G1, S, and G2M are compromised, damaged, and abnormal cell bypass these checkpoints and proliferate uncontrollably, which is another word of it's not checking the processes, and these mutations are causing uncontrollable cell growth, mutation resistance, and stuff like that. Mutant cells can replicate and form populations in generally genetically unstable cells. These cells accumulate additional mutations, further driving the cancer progression. This causes tumors, unchecked cell division, and accumulation of mutations eventually lead to I can't say these words, sorry. Angiogenesis, which is basically the formation of new blood cells to supply the tumor. Invasion into surrounding tissue, metastasizing, which is to distant organs, which basically means it's spreading. The TP53 mutation are associated with many types of cancers, lung, breast, and colon. For example, these mutations often correlate with poor prognosis, resistant to chemotherapy, and radiation, as these treatments rely on the P53 medicated apoptosis, which means cell death. These treatments expect the targeted cells to die, and when the P53 protein isn't activated, that is lost, and sometimes chemotherapy and radiation aren't effective. Another classification for mutations is induced mutations, which are caused by environmental factors. The example I'll be using for this one is the UV-induced P53 mutation in the skin, leading to skin cancer. UV radiation from the sun is our most carcinogenic environmental factor, and this happens because the UV radiation from the sun penetrates our skin and causes damage to our cellular DNA. This damage primarily occurs by forming pyrimidine dimers, such as cyclobutane pyrimidine dimers, short is CPS, and 6-4 photoproducts. These are covalent linkages between adjacent pyrimidine bases, thymine and thymine, in the DNA strand. When our DNA detects damage, the cellular machinery detects UV-induced DNA damage, and the ATM and ATR proteins are activated in response to the DNA strand breaks or damage. These proteins signal the P53 pathway to pause the cell cycle and inhibit DNA repair. DNA repair is attempted. The nucleotide excision repair (NER) pathways attempt to remove the pyrimidine dimers and repair the damaged DNA. Proteins like XPA, ERCC1, and DNA polymerase are involved. If the DNA damage is minor, repairs proceed successfully and cell cycle continues. Persistent or unrepaired damage, on the other hand, in the case of excessive UV exposure, affects the NER mechanism. Some pyrimidine dimers persist, which means they can't be changed or repaired, and DNA damage is critical for the genes like the TP53 and can lead to mutations during DNA replication. The UV-induced mutations in the TP53 often occur at specific hotspots, such as the codon 245, 248, 273, and 242. 282. These mutations are typically C to T, tyrosine to thymine transitions, or CC to TT, TETM mutations, at this pyrimidine sites. These signature mutations are directly linked to UV exposure. Because of these mutations, normal P53 functions are lost, and then the DNA can no longer bind to DNA properly to regulate target genes. The cell cycle is not halted, sorry. DNA repair genes are not activated. Apoptosis is not triggered in severely damaged DNA. In this slide, we're going to be talking about the effects on function. The first one is loss of function, um, which is the reduced protein activity. An example of this is the BRCA1 mutation and breast cancer. We can talk about the gain of function, which is a mutation that enhances or changes the protein activity. An example of this is the EGFR mutation in lung cancer. And another one, and lastly, is a dominant negative mutation, which interferes with normal protein function. An example of this is the COL1A1 mutation in osteogenesis imperfecta. Why are these classifications essential? The first one is medical diagnosis and treatment. It helps to guide genetic testing and therapy. This looks like gene therapy, for example, spinal muscular atrophy, where the SMN1 gene is affected. I have a little diagram here talking about CRISPR and immunotherapy, where the first step is to isolate the cells that are infected from the host, which is the patient. In this case, the T cells are isolated. The T cells are then engineered to express the SARS that recognize, let's say in this example, cancer. And then the fourth step is modifying the T cells and grow and expand a culture, so you have enough of them to target the site of interest. Step 5 is to the modified T cells are infused into patient, which is the host. And in theory, what's supposed to happen is that these cells attack the damaged cells at an extreme rate that all of, or most of all, or all damaged DNA is replaced by the new altered modified T cells, which track down and kill those cancerous tumors and stuff like that. In this case, this is helpful to understanding diseases and mechanisms, to understand the APC gene frame shift mutations and colorectal cancer, for example, and advancing biotechnology, enabling genetic editing. Knowing what these mutations target allow us to understand how the sequence functions using CRISPR. That's the example I use here in this diagram, CS9 for creating and correcting mutations. These classifications are essential also because they help us understand the genome and what affects it. In conclusion, gene mutations and their classifications provide an insight into the impact of genetic alteration and how that affects the function, protein expression, protein resistance, function. These examples demonstrate the relevance in health, diseases, and innovation. And lastly, the one thing that everybody should take away of these classifications is that without classifications, we don't understand and we don't have a frame to look back for reference and understand why these mutations happen, how they affect the host, and how can we take course of action when regulating these mutations. Again, this is very important in the medical field and biotechnology, as they both kind of link together to help people and other, not only humans, but other creatures in this world.